There are three anatomic types of muscle: skeletal, cardiac, and smooth. Both skeletal and cardiac muscle are classified microscopically as striated because of their stripy appearance. Skeletal muscle is also referred to as voluntary muscle because it remains relaxed in the absence of nerve stimulation. In contrast, cardiac and smooth muscle can function without nerve input and are referred to as involuntary. And we'll be talking more about those types of muscle in later sessions. The goals of this session will be to get into skeletal muscle and talk about the fundamental structural components in the functional unit we call the sarcomere, and also to talk about the molecular interactions between muscle filaments that allow for this remarkable specialization of force development. We'll see in a while when we talk about excitation-contraction coupling that the generation of action potentials in the skeletal muscle cell membrane, or sarcolemma, triggers a sequence of events inside the cell that result in force development. This ability to generate force results from the presence of specialized motor proteins inside muscle cells. From a structural standpoint, muscle is kind of like a series of cylinders within cylinders. If you look end-on at the cross-section of a whole skeletal muscle, you can see it consists of muscle columns, which are formed from bundles of muscle cells. Muscle cells are also called fibers or myocytes, and they run up and down the whole length of that particular muscle. The muscle cells are multinucleated, bounded by a sarcolemma, the name given to the plasma membrane. Inside each myocyte, there are more cylindrical structures called myofibrils. These in turn are made up of filament proteins. The myofibril arrays display a distinctive pattern of light and dark bands under the light microscope. This striated appearance arises from the orderly arrangement of structural and contractile proteins. Each repeating motif in this striated pattern is called a sarcomere. From a descriptive point of view, a sarcomere has the following elements. It is bounded at each end by a Z-line, sometimes also called a Z-disc. Extending from the Z-disc, we have thin filaments, which are composed of actin, tropomyosin, and troponins. These projecting here from the Z-line or Z-disc towards the center of the sarcomere. Towards the center, we have the thick filaments, which we'll see are composed of the protein myosin, and these thick filaments are overlapped by the thin filaments here. The sarcomeres line up end to end within a single myofibril. The darker areas that can be seen microscopically are called A-bands. The A-band here corresponds with the location of the thick filaments. Lighter areas at the ends of the sarcomeres are denoted I-bands, and they let more light through in microscopy because these are areas where there is no overlap between the thin and thick filaments. We take a look at the molecular components of the sarcomere in a bit more detail. The thin filaments have three main components. The backbone is formed by a double-stranded helix of actin. The helical groove on the actin filament is then occupied by a molecule called tropomyosin, which at rest obscures binding sites between actin and myosin. Skeletal muscle contraction is regulated by a protein complex that consists of this tropomyosin molecule plus its attached troponin subunits, shown here in green. You can see there are three of them in this trimer. There is troponin T, which anchors the whole trimer to tropomyosin, troponin C, which is the critical calcium binding subunit, and troponin I, which serves to inhibit interactions between actin and myosin when intracellular calcium is low. Down at the bottom here we see the thick myosin filament, and the myosin molecule is the molecular motor responsible for force development. It consists of a head, and the head is this critical area which consists both of the actin binding site plus all the elements necessary to bind and hydrolyze ATP for force development. The myosin heads are cross bridges that bind to actin during muscle contraction. The myosin heads are connected to the tail of the molecule via the hinge region here, and the hinge allows movement of cross bridges, which is the basis of force development. The myosin tails then are bundled together to form the core of the filament. There are some other molecules not drawn in this picture that are important. The protein titin is important for maintaining sarcomere structure, and it runs from the Z disc to the M line at the center of a sarcomere. It's a very extensible molecule, and it's largely responsible for the passive tension that is measured when a relaxed muscle becomes stretched, and the concept of preload that we'll talk about extensively in muscle mechanics and also in cardiac physiology. It's kind of the elastic spring of the muscle. Another very important structural scaffolding protein is dystrophin, which is located between the sarcolemma, the main cell membrane, and the myofilaments. Got its name because it is the mutation target in the disease Duchenne muscular dystrophy. Duchenne's is an X-linked disease that causes progressive muscle weakness and most commonly will result in death from respiratory failure. There are also milder mutations in the dystrophin gene that result in the less severe condition of Becker muscular dystrophy. The mechanism of active force generation in all types of muscle is based on the idea that thin filaments are pulled over thick filaments by the movement of those myosin heads. In a relaxed skeletal muscle, contraction is inhibited by the tropomyosin-troponin complexes, which obscure the active site on actin and prevent cross-bridge binding. That would be shown here in position 2, with the blue tropomyosins there obscuring these actin binding sites beneath. When the muscle is stimulated, the intracellular calcium concentration increases and calcium binds to troponin C. A resulting conformational change then exposes active sites on the actin molecules. A cycle of events now shown here occurs where myosin cross-bridges bind to actin, perform a power stroke, detached, become cocked again, then reattached, and this cycle will repeat itself in the continued presence of calcium and ATP. When death occurs, ATP production by mitochondria stops, and the condition rigor mortis sets in. Rigor mortis is a stiffening of the muscles. Turns out ATP is necessary for the myosin heads to detach from the actin filaments after the power stroke, this step here. Once ATP production ceases, then the cross bridges get locked into place, which is the cause of the muscle stiffening. Returning now to the living muscle, thin filaments from each end of the sarcomere move towards the center by sliding over the top of thick filaments, causing the neighboring Z discs to approach each other. So we see increased overlap here between the thin and the thick filament. So notice then that the overall length of the sarcomere in this example has decreased, the muscle has shortened, but notice also that the A band has not changed its length. This represents the myosin molecules in the center, cross bridges grabbing onto the thin filaments and pulling them closer together over the top of those thick filaments. In the next session, we'll go back and consider the whole excitation-contraction sequence, starting with action potential in the motor nerve, reaching a neuromuscular junction.